Dear students, I Bhati Sawant Kale welcomes you to Jnan Taranga NEET CET coaching program organized by Department of Pre-University Education Karnataka. My topic is Morphology and Anatomy of Flowering Plants. Let us begin with the first slide, which is showing the main parts of the plant. In previous topic under unit 1, you already discussed about 5 kingdom classification based on phylogenetic system categorized by Robert Whittaker. In that, kingdom Plantae classified in 5 divisions. The 5th division includes flowering plants named as Angiospermae. A typical angiospermic plant is having 2 vegetative axes. Descending axis is called root system and ascending axis is called shoot system. The root system is developing from the radicle of the seed, and the shoot system is an ascending axis developing from plumule of the seed. The root system is mainly for anchorage and absorption of minerals along with the water and secretion of phytohormones, while the shoot system is conducting the sap towards the leaves and translocate the solutes which are prepared by the leaves towards the parts of the plant body. Let us begin each part one by one. To begin with the shoot system, the main branch of the shoot system is stem. It is defined as an ascending axis, positively phototropic and negatively geotropic, having nodes and internodes. From the node, the leaves, buds and flowers will arise, and the successive distance between the nodes is called internode. The main function of the stem is conduction. Apart from that, the stem of various plants are modified to serve the functions like storage, perennation, protection, support and vegetative propagation. Let us come to the stem which is modified for storage of the food. The stem is underground mainly to store the food, helps in perennation, and when the time permits, it helps in propagation. For example, the tuber of potato, rhizome of ginger, bulb of onion, corm of Amorphophilus, these are some examples of stem modifications for food storage which are below soil. For perennation, subaerial stems are runner of grass, offset of Acornia, it is a hydrophyte, stolon of jasmine, suckers of mint. Apart from perennation, if you see the aerial stem modifications, axillary bud is modified into a pointed rigid structure called thorn. For support, the bud is modified into wiry structure called tendril, and the bulbils are modified buds which are helping in vegetative propagation. So overall, the stem is an ascending axis of the plant, developing from the plumule during germination. It has nodes and internodes, the nodes with leaves, buds and flowers. Apart from conduction, the stem is underground, modified for food storage. The stem is subaerial, modified for propagation. The aerial stem modifications are for protection, support and vegetative propagation. Let us move to the next slide. The descending axis of the plant usually develops from radicle, is negatively phototropic but positively geotropic. The roots are classified as the true root and adventitious root. The true roots, in which the radicle grow down the soil, persist as primary root, giving secondary and tertiary branches with the root hairs, which gives more anchorage and absorbs maximum water and minerals from the soil, is taproot. There is another root developing from the radicle, but it is not persisting throughout the life of the plant, it is short lived, and from the point there are many branches of the root will develop, which gives less anchorage in seasonal or annual plants. Absorptive capacity is also less compared to taproots, are called fibrous roots. So, true roots are taproots, which are usually found in dicotyledonous plants, and fibrous roots are usually found in monocotyledonous plants. There are adventitious roots which are developing from other parts of the plant but not from the radicle. So, there are three types now: taproots, fibrous roots and adventitious roots. Take any root, there are four main regions. The tender root apex is always protected by a root cap. Not only protection, but it gives the lubricating passage in the soil so that root will propagate below the soil. Above the root cap region, it is the region of meristematic activity, where the cells are dividing and increasing in number. Just above that, it is a region of elongation, where the cells are increasing in length and width. Above that, the cells are modified for absorption. So, here the region of maturation will produce unicellular outgrowths called as root hairs. So, regions of roots are root cap, region of meristematic activity, region of elongation, region of maturation with root hairs, and it is also called as absorptive region. Above this, the conduction region begins, where vascular tissues are organized properly. The main function of the root system is to provide anchorage and absorb water along with mineral nutrients from the soil. Apart from that, the root also helps in secretion of phytohormones, which are necessary for the growth. Other than anchorage absorption, the roots are modified for storage, support, respiration and propagation. As I said, the taproots are modified for food storage, are, you can see in the slide, the root is modified and appear like a spindle. It is swollen in the middle and tapering on both sides. So, it is the fusiform root of the radish. There is one root which is conical in shape, whose base is broad and apex is narrow. It is found in the carrot. There is one more root whose basal portion is swollen and the apex is narrow and tapering. This is found in the beet. There is one root from adventitious group which is like a spindle but arising from the nodal regions. These are tuberous roots. Like tuberous roots, there are fasciculated roots, there are nodulose roots. So, taproots modified for food storage are fusiform root of radish, napiform root of beet, conical root of carrot. And adventitious roots modified for food storage are tuberous roots of dahlia, tuberous roots of sweet potato, fasciculated roots of dahlia and nodulose roots of leguminous plants. This is about the root modification for food storage. The roots developing from aerial parts of the plant, that is from the stem and the branches or from the buds. They are providing maximum mechanical support to the growing plant. For support, the lateral branch of the stem gives descending branches called as prop roots. In banyan tree, you will find these prop roots giving mechanical support. In beetle vine, it is a climber. To take support, each nodal region produce climbing roots. In most of the monocotyledonous plants like maize, jawar, sugarcane, the basal nodes produce adventitious roots called stilt roots. They are for mechanical support. The plants growing in saline region where soil is completely covered with the salt water, we call them as mangroves. The mangroves like Avicennia and Rhizophora, they produce the aerial branches of the root with minute openings called pneumatophores for gaseous exchange. Other than this, there is epiphytic group of plants take support of the large tree branches and they produce clinging roots having velamen tissue for absorbing moisture. There is another modification where the Cuscuta-like plant, it is a parasitic plant, achlorophyllus, yellowish in color. It survives on the host plant Clerodendron. It produces microscopic roots called Hosteria, which will penetrate till the xylem and phloem and sucks whatever they want. So, to summarize, the root is descending axis of the plant, positively geotropic, negatively phototropic, having classified as taproots, fibrous roots, adventitious roots. The region of roots are root cap, region of meristematic activity, region of elongation, region of maturation with root hair, and above that there is a region of conduction. Apart from anchorage and absorption, the roots are modified for food storage, the roots are modified for mechanical support, the roots are modified for respiration, absorption, and the roots are also modified as parasitic roots. Let us go to the next slide. These are generally called kitchens of the plant. These are green in color, dorsoventrally flattened branches arising from each node, and we call it as leaf. A typical leaf has three parts: the leaf base, the petiole, and the leaf lamina. If you consider the stem branch, there is a nodal point. From nodal point, if you consider the expanded portion lamina with the midrib, these are the veins and veinlets. Now, this point is the leaf base called hypopodium. This portion is the petiole called mesopodium, and the expanded portion having margins meeting at the leaf apex is known as epipodium. So, leaf base, petiole and the lamina with margins and the apex. The leaf without petiole is called as sessile and the leaf with petiole is called petiolate. There are two tiny outgrowths on either side of the petiole in some called as stipulate leaves. The leaves without stipules are called as exstipulate. When the leaf lamina is directly attached to the leaf base without having petiole, it starts covering the stem called as sheathing leaf base. In some plants, the leaf base is swollen called as pulvinous leaf base. This is regarding the parts of the leaf and related points. Come to the next part. If you consider leaf lamina of grass, which has veins, veinlets arranged parallel to each other, this is called parallel venation. When we consider the leaf lamina having midrib, the veins and the veinlets with venules in the form of network, it is known as reticulate venation. So, venation is the arrangement of veins and veinlets on the leaf lamina. If the branches are parallel to each other, it is parallel venation. If the arrangement of veins and veinlets is in the form of network. It is called reticulate venation. So, parallel venation usually found in monocot leaves and reticulate venation usually found in dicot leaves. Come to the types of leaves. The leaf having single lamina is usually termed as simple. The leaf in which the lamina gets dissected into leaflets or pinnae, then it is called compound leaf. When the leaflets or pinnae are arranged parallelly along the rachis is pinnately compound, and when the leaf lamina dissected at the apex into leaflets, then it is palmately compound. When the leaflets are arranged parallelly along the rachis, we call pinnately compound. Both are compound leaves, and when there is a single lamina, it is called simple leaf. When the leaves are produced on the nodes, there is only one aim. It has to absorb maximum solar energy to prepare food during photosynthesis. So, the specific arrangement of the leaves on the stem is known as Filotaxi. When a single leaf develops from a node, we call alternate. It gives spiral mode. If you consider a stem, from each node the leaf develops, and if you trace the path, it goes spirally. So, it is also called as spiral Filotaxi. A pair of leaf developing from a node is known as opposite Filotaxi. This is the stem, the node, and these are the paired leaves. This is opposite Filotaxi. There is one more type where the stem from the node there are many leaves develops, forming a whorl around the node. This is whorl Filotaxi. So, one node, one leaf, alternate, gives spiral arrangement. So, it is called alternate or spiral Filotaxi. A pair of leaf from single node, it is opposite Filotaxi, and many leaflets from each node is known as whorl Filotaxi. The main function of leaf is to prepare food by photosynthesis, but apart from that, the leaves are modified for various purposes. According to the syllabus, the leaf modified for propagation is leaf bud. The leaf modified for protection is a protection as well as to conserve the water in xerophytes is the spines. The leaf is modified into wiry structures called as leaf tendrils. It is usually found in Pisum sativum, garden pea plant. There is pitcher plant called Nepenthes. There is bladderwort called Utricularia. There is Drosera or sundew plant. In all these are called usually insectivorous plants. In all these plants, the leaves are modified to trap the insects to derive the nitrogenous compounds. So, to summarize, the leaf is dorsoventrally flattened green part of the plant developing from the nodal region, having leaf base, petiole and leaf lamina. Lamina shows parallel venation and reticulate venation. Leaves are classified as simple leaf, compound leaves. Compound leaves with pinnately arrangement and palmately arrangement. Filotaxi is of three types: alternate, opposite and whorled. And other than photosynthesis, leaves are modified for propagation, support and protection. Coming to the next slide, a special floral axis on which the flowers are arranged is known as inflorescence. So, in simple language, it is a bunch of the flowers or typical arrangement of the flowers. In the evolutionary process, this inflorescence provide maximum approach for the insects to pollinate them. So, inflorescence is the evolved character in most of the plants. If you see the diagrams, there is racemose inflorescence where the old flowers are at the base of the peduncle, and young flowers are towards the apex, and the apex is continuously growing. This kind of inflorescence we call racemose. It is indefinite because rachis growth or peduncle growth is continuous. So that old flowers will remain at the base and young flowers go on developing towards the apex. But here the tip of the rachis never ends into flower. If you look at the another diagram where the peduncle ends into oldest flower and the young flowers are below the old flower. So here it is definite kind of inflorescence where rachis growth is not continuous, terminating into a flower which is oldest, and below that young flowers are growing. This kind of development we call basipetal, and such inflorescence we call it as cymose inflorescence. So racemose inflorescence where the rachis growing continuously with acropetally arranged flowers, and cymose inflorescence where flowers arranged basipetally and the rachis terminates into a flower. The flower is the highly modified condensed axis for sexual reproduction. To begin with the flower usually develops in the axil of a small leaf, and that we call bract. The flower with bract and the flower without bract. The flower having two groups of whorls arranged cyclically one above the other, supported by a tip of the pedicel called thalamus. On the thalamus, the first whorl is usually green in color, known as calyx. The second whorl is usually colorful, known as corolla. The third whorl is coming under essential, known as androecium, called as male whorl. And the fourth whorl is also essential, necessary for reproduction, called as gynoecium. When the flower is having calyx, corolla, androecium and gynoecium, we are calling it as complete flower. And when the flower is either with androecium or with gynoecium, we call unisexual flowers or incomplete flowers. Flowers having both androecium and gynoecium together, we call it as bisexual flowers. The flowers in which a central division plane dividing flower in two equal parts, then it is zygomorphic symmetry. A cut passing through any plane dividing the flower in equal parts, it is called actinomorphic symmetry. Actinomorphic and zygomorphic, these are the two regular symmetries in most of the flowers. The flowers cannot be divided into two equal parts in any plane, it is known as asymmetric flower. Flower is always supported by a stalk known as pedicel. The flower with pedicel is known as pedicellate, and the flower without pedicel is called as sessile. Sometimes below the calyx there are many green outgrowths called as bracteoles. So flowers with bracteoles and flowers without bracteoles. The flowers in which each whorl having 3 members are called trimeres. The flowers in which each whorl is having 4 members called as tetrameres, and the flowers having the whorl with 5 members called as pentameres. So generally flower is defined as highly condensed modified axis of the shoot for sexual reproduction. They are pedicellate or sessile, bracteate or ebracteate, bracteolate or ebracteolate, unisexual or bisexual. Unisexual we usually call staminate, male flowers, and pistillate are the female flowers, and there are some flowers which remain sterile called neuter flowers. The flowers having regular symmetries like actinomorphic and zygomorphic, and the flowers having irregular symmetry known as asymmetric. The flowers having the definite number of members in each whorl, accordingly we call it as trimeres, tetrameres and pentameres. And when you go to the next slide, we can see the thalamus on which a special part of gynoecium is ovary. It is a site for megasporogenesis, in which ovules developing having the female gamete inside. It is a site for sexual reproduction, and it is a site for embryonic development, plus it is a area which develops into fruit. So, it is highly protected and highly supported. So, if you see the diagrams, there are three positions of ovary. The first diagram where ovary is above all, below the ovary remaining parts are arranged. So, this kind of arrangement we call ovary is superior and flower is said to be hypogynous. The second diagram shows the ovary is, and second and third diagram shows ovary is half inferior and half superior. It is at the rim of the thalamus. So, we call it as perigynous flower. The last diagram showing the thalamus is like a flask shaped. Ovary is highly protected inside and remaining whorls are arranged above that. This kind of arrangement is found in epigynous flowers. So, ovary with superior ovary is the flower with superior ovary is hypogynous. The flower with half superior half inferior ovary is called perigynous. The flower with inferior ovary is called as epigynous. When you consider the arrangement of sepals and petals with respect to each other in calyx and corolla is known as estivation. The first diagram, in which the members are having the margins just touching to each other without overlapping and overlapped, it is known as valvate estivation. The second diagram shows the margins of each member is overlapped and overlapping, it is twisted estivation. The third diagram is showing the margins of each member are not regularly overlapping and overlapped, it is known as imbricate. And the fourth diagram is very special, it is usually found in papilionaceous corolla of leguminous plants. In this, the standard petal Xylem with overlapping margins, the two wing petals are with overlapped and overlapping margins, and two smallest petals whose margins are fused below and other margins are overlapped by wing petals. This kind of estivation is known as vexillary estivation. When you take transverse section of ovary, we can see the arrangement of ovules on the placenta. When the ovules are arranged along the margins where both margins of the ovary are fused, it is known as marginal placentation. When the ovules are arranged on the central axis of the ovary, we call it as axial placentation. When the ovules are arranged along the periphery of the ovary wall, it is known as parietal placentation. And when the ovules are arranged in the center part, freely available, they are called free central. In monocarpellary condition, the ovule is single attached at the base of the ovary, it is known as basal placentation. So, marginal ovule along the margin, axial ovules on the central axis, parietal ovules along the periphery, and free central freely in the center of the ovary, and basal at the base. The product of sexual reproduction is the fruit. The fruit developing from ovary we call true fruit. Sometimes ovary fails to develop into fruit, then some other parts of the flower develops as fruit, then we call it as false fruit. And the fruit will develop without fertilization, we call it as parthenocarpic fruit, which remains seedless. In diagram there are two fruits showing the pericarp. It is a fruit wall differentiated in three layers: epicarp, mesocarp and endocarp. In mango, the epicarp becomes leathery and it appears as yellowish in color. The mesocarp at maturity remains sweet and it gives the pulp, it is edible part. The endocarp is stony in mango and the seed is single. In another diagram, a coconut whose epicarp is peppery, mesocarp is fibrous, endocarp is stony, and here endosperm is edible, having endospermic fluid. Both are true fruits. they comes under drupe. Let us come to the next slide. There are two types of seeds shown on a slide. One seed is monocotyledonous and other seed is dicotyledonous. In monocotyledonous seed, the example is maize. The lower portion is embryonal part of the seed and the upper portion is the endosperm. This seed is with endosperm, while here you can see two cotyledons, embryonal axis with anterior plumule and posterior radical without endosperm. This is non-endospermic. Endospermic seed is having a single cotyledon, it is called scutellum. In non-endospermic exalbuminous seed, the two cotyledons remains fleshy. Here, the plumule has a protective sheath coeloptile and the radical has protective sheath coelorhiza. There is alluron layer having a protein around the endosperm. This is the epithelial layer separating endosperm from embryonal part. Around alluron layer, it is a pericarp. The pericarp means fruit wall and the seed coat, outer covering of the seed. In this, fruit wall and seed coat cannot be separated. This kind of fruit we call it as Karyopsis. In this diagram, it is a diagram of dicotyledonous seed, non-endospermic exalbuminous, having two cotyledons, embryonal axis with plumule and radical covered by seed coat. There is a point where hilum, this hilum is a scar remain on the seed where the funicle was attached to the fruit. Below that there is a small opening from which the germination begins called as micropyle. Look at this diagram carefully. One by one we will study the technical terms to describe the flower. To begin with, the flower is having a small leaf. In its axil, the flower is growing. So that small leaf is known as bract. Flower with bract is termed as bracteate. It is written as BR in floral formula. Without bract, it is technically written as EBR, means ebracteate. There are some green outgrowths called bracteoles. If bracteoles are present, it is written as BRL and it is termed as bracteolate, and without, it is EBRL, it is known as ebracteolate. The flower is complete, having regular symmetry. If the line is drawn in the center, it is zygomorphic symmetry. It is having regular symmetry, and inside the symbol of plus is shown, it is actinomorphic symmetry. When we come to the next part in floral diagram, so it is graphical representation of floral parts. Here, the outermost is the calyx, the members are sepals. It is written as K. In this diagram, there are 4 sepals are shown. So I can write here 4. Here, estivation, if you see, they are not in proper. There are 2 members with overlapping and there are 2 members with overlapped margins. But here sepals are free. So there is no need to put bracket around this number. If, for example, calyx having 5 sepals and they are fused, it is known as gamosepalous, and if you show this bracket around the number, it indicates that calyx is gamosepalous, without bracket calyx is polysepalous. The next non-essential whorl or accessory whorl is corolla. The members are petals which are brightly colored. Usually they attract the insects for pollination. They have nice fragrance. The calyx having 5 petals. They are not fused, we call polypetalous. Calyx having 5 petals and they are fused, we call it as gamopetalous. A stands for androecium. In androecium, if single stamen or many stamens, that number is written here. For example, there are 5 stamens. G is 5 carpels. Androecium in which out of 5, 3 are fused to form one bundle and remaining 2 are free. So, there are 2 bundles called as diadelphi. When all these are united, it is adelphi. So, adelphi showing one bundle monoadelphi, adelphi showing 2 bundles diadelphi, adelphi showing many bundles of stamens called polyadelphi. Gynoecium, where when the line is drawn below, it is hypogynous flower having superior ovary. Gynoecium, where ovary is inferior, the flower is epigynous. Gynoecium, ovary is half inferior, half superior, flower is said to be perigynous. The respective members will specify the number of carpels. When they are free, we call apocarpus. When they are fused, we call it as syncarpus. Look at the floral diagram given below. It is actinomorphic symmetry. The flower is bisexual. This is the flower staminate having only androecium. The flower having only gynoecium are pistillate. The flower having androecium and gynoecium are bisexual. Respective arrangement is shown below. Let us move to the next slide. Family Fabaceae or Papilionaceae, these are subfamilies of Leguminosae. These are herbs or shrubs, usually climbers. Leaves are modified into tendrils for the support. The floral characters, if you see, the calyx is gamosepalous. The corolla is papilionaceous with a axillary estivation. If you look at the floral diagram, it is zygomorphic. The sepals are showing valvate estivation. Corolla is showing axillary estivation. These are the stamens, diadelphous 9+1. The ovary, the gynoecium is monocarpellary with basal placentation. These are pulse yielding families, oil yielding families, or dye yielding plants are included in this family. This is potato family Solanaceae. These are herbs or shrubs. The stem is usually branched. The roots are tap roots. The leaves are simple petiolate. They usually show cymose type of inflorescence. In earlier slide, they show racemose type of inflorescence. In Solanaceae, if you look at the flower, it has 5 sepals. Look at the floral diagram, 5 sepals with valvate estivation, 5 petals with valvate estivation. Symmetry is actinomorphic. There are 5 stamens which are not fused but they are attached to the petals. So, it is epipetalous condition. There are bilocular condition in the ovary as it is bicarpellary, and ovules are on axial placentation. It is lily family, known for the bulb or rhizome. It has fibrous roots. The leaves are basal in position and they are culline type with parallel venation in the leaf lamina. The diagram shows umbel type of inflorescence where the floral parts, if you see here, calyx and corolla are fused. They are not distinct, so we call it as perianth. The members of the perianth are called tepals and they are arranged as 3+3. The androecium with stamens attached to the tepals, so it is called epiphylous condition. Tricarpellary condition in the ovary with axial placentation. After this, the brief session, let us start with some questions. The stilt roots in maize are modified for Definitely not for food storage, but stilt roots are arising from most of the monocotyledonous plants from the basal node for support. They are not helping in propagation, they are not helping in gaseous exchange. So, the correct answer is support. In racemose inflorescence, flowers are definitely not at the apex because this kind of arrangement is in cymose. Acropetally arrangement, yes, because basal flowers are old and young flowers are towards apex. Basipetal arrangement is in cymose and centripetal arrangement we found in the head or capitulum. So, the correct answer is the flowers in racemose inflorescence are acropetally arranged. In the given diagram, the position of ovary, if you see on the thalamus, it is at the rim of the thalamus, where the attachment of stamens, attachment of calyx and petals and the ovary is at the equal length. So, this kind of position we call half inferior, half superior. So, the correct answer is perigynous flower. Moving to the next slide, already we have described, we have learned that the arrangement of sepals and petals with respect to each other in calyx and corolla is called as estivation. A diagram is showing valvate estivation where margins are not overlapping and overlapped. B diagram is showing where the members having the margins with overlapping and overlapped. Third diagram where the margins of the members are not regularly overlapped or overlapping, and in the fourth diagram very special with vexillary estivation having standard petal with overlapping margins, wing petals with overlapping and overlapped margins, and the keel petals are with overlapped margins. So, the correct option is A. If you look at the placentation, as I said earlier, when the ovules are arranged along the periphery of the fruit wall, it is said to be parietal placentation. So, correct answer is parietal placentation. An example of axial placentation is marigold, argemone, they have basal placentation. The lemon has axial placentation. So, the correct answer in this is the lemon. Leaves are modified into spines in. So, if you look at the options, the pea, onion and silk cotton, these are the mesophytes. They grow where the water is abundant. The Aponcia is a plant adapted for xeric condition, where the stem is modified as Phyloclade and the leaves are modified into spines. So, correct option is Aponcia. The hilum is a scar on the seed where the funicle was attached. This I said earlier when we studied about the fruit as well as the seed. So, the correct answer, the hilum is a scar on the seed where funicle was attached. The fruit developing from ovary is called true fruit. When the fruit remains fleshy at maturity, we call it as fleshy fruits. When they become dry, we call dry fruits. But fleshy and dry fruits, they all are developing from polycarpellary syncarpous gynoecium. But when you see the etario, the bunch of the fruitlets, it is said to be an aggregate fruit, and this aggregate fruit is developing from multicarpellary. Carpels are many, but they are not fused. So, when they are not fused, we call apocarpus. So, the correct option is multicarpellary apocarpus gynoecium. The cotyledon of maize grain, it is a seed with endosperm having embryonal axis with plumule covered by coeloptile, radical covered by coelorhiza and having a shield shaped single cotyledon known as scutellum. So, correct answer is scutellum. The standard petal of papilionaceous corolla is also called, there are 5 petals in papilionaceous corolla. A standard is the largest among all called Xylem. The two are lateral, they are called wing petals, and the two are smallest are carina or keel. The correct option here is the Xylem. When margins of sepals or petals overlap on one another without any particular direction, definitely it is a kind of imbricate estivation. So, the correct option is imbricate. Pneumatophores are the special aerial root branches developing in the plants called mangroves, and there are two examples of mangroves, Rhizophora and Avicennia. So, the option shows Rhizophora, and these pneumatophores are helping in gaseous exchange, that is respiration. So, it is found in Rhizophora. In Bougainvillea, the leaves are modified into pointed structures called thorns. It is an aerial modification of the stem, not the leaf. These thorns will give protection to the plants from grazing animals. So, Bougainvillea thorns are the modifications of stem. This floral diagram is showing actinomorphic symmetry, 5 sepals with gamosepalous condition, 5 petals having stamens attached to it called epipetalae, bicarpellary ovary. This kind of arrangement we found in the flowers of Solanaceae. The correct option is Solanaceae. The leaf tip tendrils are present in Pisum, the leaf is modified into tendril, but in Gloriosa superba, the leaf tip is modified as tendrils for mechanical support. In spiral Philotaxi, the number of leaves, when single leaf is developing from each node, we call alternate. When two leaves in opposite direction develops, we call opposite Filotaxi, and when there are many leaflets arising from a single node, we call it as whorled Philotaxi. So, when you trace the pathway of alternate Filotaxi, it gives a spiral arrangement. So, the correct answer is a single leaf is present from each node. So, correct option is 1. There are 1, 2, 3, it specifies the number of leaves arising from each node along with many. So, if you consider a stem, a leaf single arising from each node, and if you trace its path, it gives spiral arrangement. So, it is spiral Philotaxi. These are two leaves in opposite direction. When two leaves in opposite direction arranged one on the other, this is opposite superposed. In Calotropis, this is found in Quiscalis indica, Rangoon creeper. Here, the node giving a pair of leaves, this is in Calotropis, these are sessile leaves. There is another pair right angle to the first. So, first pair and second pair both are making right angle to each other. It is opposite decussate, opposite superpose, opposite decussate, and this is alternate showing spiral Filotaxi. When the node showing many leaves arising, forming a group around this is known as whorled Filotaxi. So, in spiral Filotaxi, the number of leaves at each node is 1. Which of the following is not a stem modification? Flattened structure of Opuntia, these are called Phyllochletes. They are mainly for photosynthesis because here leaves are modified into spines. The tendrils in cucumber, these are the stem modifications for mechanical support. Thorns of citrus plants, these are modified buds for protection. So, these three are stem modifications. So, the left is the picture of Nepenthes where the leaf base with the picture having hydrolytic enzymes and sensory hairy structures, and the tip is having a lid. The lid is attractive in color. This ostiole the plant keeps open. Due to beautiful color of the lid, the insects are attracted. As they come here, they are trapped by sensory hairs. Once the insects are falling in the pitcher, they are not allowed to go out, which are crushed and nitrogenous food is derived from it. Other than Nepenthes, there is a bladderwort. It is maximum found in the Western Ghat area, and there is another plant called sundew plant. In all three plants, the leaves are modified to trap the insects to derive the nitrogenous compounds. Let us move to the next slide. The stem modified into flat green organs performing the functions of leaves are known as cladode, phylode, phyloclade or scales. Just before that, I told you Opuntia is having a flat stem on which the spines are arranged. These are modified leaves to reduce the water loss during transpiration. So, as the leaves are modified into spines, the stem has to start performing function of photosynthesis. So, these stems are remaining green and flat, having succulent tissue inside, and these are called Phylloklades. In family Liliaceae, the three carpels are there. All are fused, so it is syncarpous condition, and this is a characteristic of, it is a trimerous flower and is a characteristic of a lily family, commonly known as lily, but the scientifically we call it as Liliaceae. So, the correct option is Liliaceae. The vascular bundles in dicot stem, okay, this I will be explaining. When xylem and phloem are along the same axis and they are together, we call it as conjoined collateral, where protoxylem is towards pith and metaxylem is towards pericycle. It is said to be endark xylem. And when xylem and phloem is separated by intrafascicular cambium, it is known as open. So, in dicot stem, vascular bundle shows conjoined, collateral, endark and open condition. If you look at the two columns, dicot stem, monocot stem, dicot root, monocot leaf. Okay, this we are learning. Sure, monocot leaf with bulliform cells, dicot root with Casparian strips in the endodermal region, monocot stem with conjunctive tissue to store the water and the food, dicot stem with star sheath. These are the columns given. Here the odd is the conjunctive tissues are the water transporting or water storing tissues. They are not found in the stem, they are in the root. So, the odd pair is monocot stem and conjunctive tissue. Dear students, so far we have learnt the external parts of the flowering plants. We call it as morphology of flowering plants. Let us go inside the flowering plants to see how the cells are arranged layer wise, and we can compare with monocot and dicot. So, the next session is anatomy of flowering plants. To learn anatomy, we should know about the plant tissues. Basically, plant tissues are classified into two groups, meristematic and permanent. The basic difference is meristematic tissue is having embryonic cells capable to undergo mitotic divisions, and hence they are responsible for the growth. Permanent tissues are derived from meristematic tissue after differentiation. They lack ability to undergo mitotic division, but they are mature and they perform various functions in the plant body. Let us begin with the meristematic tissue. They are classified based on the origin, based on the position, based on the function and based on the plane of division. Based on the origin, there are two meristematic tissues, primary and secondary. The primary meristematic tissue is located right from the birth of the plant, and it remains embryonic till the death of the plant. So, the growing regions of the plant are always have primary meristems, and they are usually at the root and shoot apex and at the nodal regions. The secondary meristem is developing by dedifferentiation, where the permanent tissues like parenchyma, they attain ability to undergo mitotic division, and they will give rise secondary meristem which redifferentiate as secondary tissues. So, secondary meristem is exclusively for secondary growth, and secondary growth is usually found in dicotyledonous plants as they are perennial. Secondary growth is usually absent in monocots as they are seasonal annual plants. The meristem is located at the shoot or root apex, known for linear growth of the plant body, is known as a apical meristem. The meristem present at the nodal region responsible for the development of node, the leaf, the bud, the branch, the flower is known as intercalary meristem. The meristem located along the periphery, usually secondary in its origin, is known as lateral meristem. The meristem differentiate giving outermost protective layer is known as protoderm. So, protoderm will give rise epidermal tissue system. Below epidermal tissue system, there is a mechanical layer hypodermis. There is a layer called cortex, there is a layer called endodermis, there is a layer called pericycle along with the pith. These peripheral layers are derivatives of ground meristem. The special conducting tissues, we call it as vascular tissues, are xylem and phloem, and they are derivatives of procambium. The permanent tissues are divided in two groups. The simple tissues having the cells with same origin, same structure, sometimes dissimilar structure, but perform a same function. We call them as simple or homogeneous tissues. In this, the most fundamental tissue found in almost all layers of the plant body is called as parenchyma. The tissue which is absent in the root, present only in the aerial parts of the plant, giving tensile strength, known as collenchyma. The tissue which is having dead cells, hard cell walls, known for mechanical support and protection, known as sclerenchyma. Parenchyma, collenchyma, sclerenchyma, these are simple permanent tissues. The tissues having different types of cells, and they work as a unit for conduction. So, there are two complex tissues which are we call heterogeneous. They are xylem and phloem. Xylem is known for water conduction and phloem is known for translocation of the solutes. To begin with anatomy, we should know three tissue systems. The epidermal tissue system comprises a single layer, and that we call it as epidermal tissue system. If you consider root, it has thin walled cells, non-cutinized, and they will produce the unicellular outgrowths. These are called root hairs. This layer is characteristic feature of the root, known as piliferous layer or epiblema, non-cutinized, with the unicellular root hairs for absorption. In stem and leaf, the parenchyma cells are barrel shaped, compactly arranged without intercellular space and usually covered by a cuticle for extra protection. This is epidermis. In dicotyledonous plants, epidermis of young stem and the leaves will produce multicellular outgrowths. These are called trichomes, which are usually absent in monocot stem and the leaf. In leaf epidermis, the cells are modified like bean shape, leaving minute opening known as stoma. There is another kind of shape, looks like a dumbbell, having a stoma. The bean shaped guard cells, dumbbell shaped guard cells enclosing stoma for transpiration. These epidermal openings we found in the leaves for transpiration. So, bean shaped stomata is characteristic of dicot, and dumbbell shaped guard cells is a characteristic feature of the monocot leaves. Below this, there is a ground tissue system. Ground tissue is having a mechanical layer, hypodermis. It is having a multi-layered parenchymatous cortex. It is having single parenchymatous layer known as endodermis. Hypodermis, cortex, endodermis, these are peripheral layers below epidermis. Below endodermis, it is pericycle, and the central axis is pith with extend as medullary rays. This portion we call stele, and stele has a border of pericycle. So, hypodermis till pith, these all are comprising ground tissue system. In the stele region, the procambium differentiate to give vascular tissues. The vascular tissue is xylem and phloem. When xylem and phloem are arranged separately, we call it as radial bundles. When they are together, we call conjoint. Along the same axis, we call collateral. Separated by cambium, it is open, and without cambium, it is closed. There is latex yielding group of plants. In that, the vascular bundles are bicollateral, where xylem is sandwiched between cambium and phloem. There are some plants where vascular bundles are concentric, where xylem is surrounded by phloem or phloem is surrounded by xylem. So, it is generally called amphicribal and amphivessel respectively, and they are called as concentric vascular bundles. So, epidermal tissue system is composed of epiblema with root hairs in the root, epidermis with cuticle in the stem and the leaf, having trichomes. It is a characteristic feature of dicot leaf and stem. Stomata are epidermal openings usually found in the leaves. Below epidermal tissue system, hypodermis for mechanical support, cortex for storage and transport, endodermis, it is having a specific function in the root and the stem. Then pericycle, it may be parenchymatous or sclerenchymatous, and the pith is a central parenchymatous axis extended as medullary rays to separate vascular bundles. The vascular tissue systems comprises xylem and phloem, having radial arrangement, conjoint collateral open, conjoint collateral closed, bicollateral or concentric with amphivessel and amphicribal. This is the arrangement of xylem and phloem. The first diagram where xylem and phloem are separated and they are differently arranged on the radius. It is radial type of vascular bundle. It is found in monocot and dicot root. The second diagram is showing xylem and phloem without cambium. This arrangement is conjoint, collateral, closed. And here the third diagram where xylem and phloem are together, conjoint along the same axis, so that it is collateral with cambium, so it is open. So, root having radial vascular bundles, monocot stem having conjoint collateral closed vascular bundles, dicot stem is having conjoint collateral open vascular bundles. Let us begin with the different sections passing through root and stem. Look at the diagram showing monocot stem and dicot stem. See the ground plan of both the stem. Here it is epidermis, no trichomes are found. Hypodermis is the second line, it is usually sclerenchymatous. You compare this ground plan with dicot, where epidermis with trichomes, hypodermis is cholenchymatous. Here ground tissue remains undifferentiated in monocot, while in dicot ground tissue differentiated as hypodermis, cortex, Endodermis is usually called starch sheath as it stores starch granules, and pericycle is sclerenchymatous, it is wavy in appearance. If you look at the pith region, it is very big in dicot stem. There is no pith differentiated in monocot. If you look at the vascular bundle, they are scattered in monocot, it is atactostele, and here conjoined collateral open bundles which are wedge shaped, arranged in proper broken ring like structure, it is known as eustele condition. If you see the enlarged section of dicot and monocot, epidermis with trichomes, collenchymatous hypodermis, parenchymatous cortex, starch sheath is endodermis. This is pericyclic patch supporting vascular bundle like a bundle cap. This is primary phloem, cambium which is intrafascicular, and then metaxylem, protoxylem. Here protoxylem is towards pith, so it is said to be endarc. Metaxylem is towards pericycle. In monocot stem, epidermis without trichomes, hypodermis sclerenchymatous, ground tissue undifferentiated, and each vascular bundle is conjoined collateral closed, surrounded by bundle sheath, and here metaxylem, protoxylem which is inactive, so it is a lysigenous cavity and it is surrounded by a bundle sheath. Okay, these are conjoined collateral closed vascular bundles. Look at the two roots of dicot and monocot. If you see outermost layer epiblema with root hairs, it is common. Here hypodermis is not found. As the roots are below soil, they do not require mechanical support. So, you do not find hypodermal region in the root, but cortex is well developed. Here, if you look at the endodermal region, it is shown in pink color. These are the cells non-suberized, and there are some cells having thick walls. Non-suberized are passage cells and suberized are Casparian strips. These are adjacent to xylem and these are adjacent to phloem. So, endodermal region of the root is with passage cells and the Casparian band cells. If you look at the number of vascular bundles, they are radial in nature. They are 4 xylem and 4 phloem in dicot root and many xylem and many phloem in monocot root. Dicot root is said to be tetrarch and monocot root is said to be polyarch. Tetrarch 4 bundles, polyarch many bundles. Pith is slightly, stele is slightly bigger, cortex is reduced, stele is small and cortex is big. These are some differences between dicot root and monocot root. Look at the two diagrams of the leaves. If you practically visualize the position of the leaves on the stem, the leaf of the maize always grow parallel to the stem. So, naturally it receives equal amount of sunlight on both the surfaces. So, the color of the leaf remains same on both sides. And if you look at the anatomy, they have stomata on both sides. So, rate of transpiration is equal. If you look at the mesophyll part of the leaf, it is chlorenchymatous where cells are loosely arranged. Photosynthesis rate and storage of the food remains same on both the sides. Such leaves are said to be isobilateral. The leaves morphologically, anatomically, physiologically similar on both the sides are said to be isobilateral, and these are usually found in monocotyledonous plants. If you compare this diagram with the dicot leaf, here the leaf arrangement, if you see, this is the stem, the leaf is making the right angle to the stem axis. So, its upper dorsal surface is towards the light and lower surface is away from the light. So, upper color is slightly dark than the lower color. Upper portion, dorsal portion receives more sunlight than the lower portion. So, naturally below upper epidermis rate of photosynthesis is more and below lower epidermis rate of photosynthesis is less. Stomata are located on the lower side to reduce the water loss during transpiration. So, these leaves are called dorsiventral. There is one more arrangement in the monocot leaf where the bulliform cells are observed only on the upper epidermis of isobilateral leaf. In adverse condition, these bulliform cells, they lose water, they shrink and they roll down the leaf lamina. This is adaptation to survive in adverse conditions. When you look at the two diagrams, epistematic condition usually in hydrophytic leaves that you do not find here. Hypostematic condition is in dicot leaf, amphistematic condition is in monocot leaf. If you look at the mesophyll, it is palisade where the cells are columnar closely arranged and spongy where cells are oval and loosely arranged with lots of intercellular spaces. So, mesophyll is palisade and spongy in dicot and here mesophyll is spongy in monocot. So, the differences are clear between monocot leaf and dicot leaf. The most important part of anatomy where usually the confusion occurs is the secondary growth. Let us take a new slide. Let us start with the secondary growth. It is characteristic feature of dicot plant. In the secondary growth, additional secondary tissues develops and it is by a special process called dedifferentiation. The result of dedifferentiation is development of secondary meristem called cambium. So, dedifferentiation will produce secondary meristem. In stem, if you consider secondary growth in stellar region, this is the stele. These are vascular bundles which are conjoined collateral open having already intrafascicular cambium. It is primary lateral meristem. During dedifferentiation in medullary region, parenchyma cells dedifferentiate and it gives interfascicular cambium. Intra and interfascicular cambium will give rise cambial ring. So, in stellar secondary growth, first is formation of cambial ring by fusion of already available intrafascicular cambium and later developed interfascicular cambium. Now, in cambial ring, these cells are fusiform initials and these cells become ray initials. Fusiform initials develop into secondary xylem and secondary phloem, while ray initials develop into secondary medullary rays. When fusiform initials undergo redifferentiation, secondary xylem develops seasonally than secondary phloem. So, there are two seasons in year. In spring season, the plant develops new leaves. So, demand of the water will increase. So, during spring season, a xylem develops known as early wood. Xylem commonly known as wood. As it develops in spring season, so it is known as spring wood. There is another season where the plants will shed their leaves, so water demand decreases. So here in this season, xylem develops with narrow lumen known as late wood. Early wood where xylem develops with wide lumen. Late wood where xylem develops with narrow lumen. Late wood is also called as autumn wood. So there are two patches, spring wood, autumn wood. Okay, so here like this, spring wood, autumn wood. Okay, so there is a complete layer with two patches developed in each year, and this we call annual ring. So every year one ring is added. So continuously this region go on adding the annual rings. So outer side the new rings are developing and old rings are deposited inside. So after many years, this portion where the xylem vessels are filled with metabolites like oil, tannin, resin, etc. Okay, so this region becomes hard, known as duramen. And outer side still the xylem is developing, where sap conducting capacity is there, it remains soft, known as alburnum. So duramen with metabolites cannot conduct sap, alburnum with the sap can conduct metabolites. Around that secondary phloem go on developing. This is called secondary growth in stellar region. So generally after many years there is a formation of duramen called heartwood, there is a formation of alburnum called sapwood. Duramen, alburnum, these are formed by continuous development of secondary xylem, which is seasonally very active compared to secondary phloem. Both are derivatives of fusiform initials, and as the secondary vascular tissues go on adding, even ray initials will add secondary medullary rays, and girth of the stele go on increasing. This is called intrastellar or stellar secondary growth. As you move further, there is extrastellar secondary growth. In extrastellar secondary growth, this is epidermis, this is hypodermis, and this is cortex. So in this region, a cambium develops, and this we call corcambium. Scientifically we call it as phellogen. Cambial ring is vascular cambium. Corcambium is phellogen. Vascular cambium for stellar growth and phellogen or corcambium for extrastellar secondary growth. Now in the corcambium, here the cells go on dividing toward outer side and they are suberized, forming phloem, and towards cortex the cells are dividing, it forms phloem. Phloem and phloem together becomes periderm. So stellar secondary growth or extrastellar outside the stele, when you see extrastellar secondary growth, corcambium is responsible, it is known as phellogen. Phellogen divides towards outer side, cells are suberized, called phloem. They divide towards inner side, the cells are non-suberized, called phloem. Phloem, phloem together constitute periderm, and along with primary tissues of cortex and hypodermis, it forms a hard region called bark. As the periderm start developing, the epidermis ruptures and cortical tissues start projecting and it appear like a lens. So the bark always show minute pores at the surface, they are called lenticels. They are with complementary tissue below. So, lenticels are usually for gaseous exchange. They may help in transpiration or they may absorb moisture during night or early morning. So, overall secondary growth, if you see in the stele, it is due to vascular cambium, and outside the stele is due to cork cambium. Vascular cambium is formed as a ring by joining intrafascicular and interfascicular cambium, where fascicular cambium will become fusiform initials, where seasonally secondary xylem becomes more active than the phloem. Usually phloem development is very slow. Secondary xylem develops in two patches, spring wood and autumn wood, forming annual ring every year. So, by counting annual rings, we can identify age of the plant. So, there is a special branch called dendrochronology which explains the mechanism to find out the age of the plant by counting annual rings. After several years, by depositing annual rings one above the other, xylem elements are filled with the metabolites and it forms a heartwood or duramen surrounded by yellowish colored alburnum. In the extrastellar region, secondary growth is due to phellogen or cork cambium, where cells are towards epidermis with suberin deposition called phloem, and the cells which are non-suberized towards primary hypodermis become phellodum. Phloem, phellodum constitute peridum. Peridum along with primary layers forming bark, and usually on the surface of bark we can see the openings called lenticels, and below lenticels we can see these complementary tissue projecting out for gaseous exchange, sometimes transpiration or sometimes absorption. This is how the secondary growth go on increasing girth, that is diameter of the plant body, and help in perennation. If you look at this diagram, it shows secondary growth in dicot root. There is no much difference in the stellar region of the root compared to the stem, but here as the radial vascular bundles are there, below phloem there is a cambial cells develops by dedifferentiation, which develops further as secondary xylem and secondary phloem. Initially it becomes a wave-like appearance and slowly it gets circular and it starts providing mechanical support. Let us come to the some questions which are there on anatomy and morphology. As I said, meristematic cells are embryonic. They have conspicuous nucleus. Yes, they are in active metabolic phase. They have developing plastids, but they do not have excretory materials. So, the correct answer is B. The simple tissue which constitute few layers below the epidermis. Okay, whenever you consider any section below epidermis, especially in the stem, okay, there is a simple tissue for mechanical support, and the mechanical tissue present only in the stem is collenchyma. So, the correct answer is collenchyma. You can easily identify now, as the vascular bundles are scattered at tactostele condition, no trichomes are seen and ground tissue is undifferentiated. So, definitely it is a monocot stem. So, option A is the correct answer. As you move further, the type of vascular bundle shown, it is very clear when xylem and phloem are separated, we call it as radial vascular bundle. When they are together, it is conjoined. When xylem is in the center, sandwiched between the phloem patches with cambium known as bicollateral, and when each vascular tissue surrounded by other, it is concentric. In the regions of a root starting from the tip upward, the four zones occurring in the following order. Recall the morphology of flowering plant, where the tip is covered by root cap, above that meristems are in division phase, above that cells are increasing in length and width, and then they start maturing, producing the root hairs, and this we call it as the D correct option. Which of the following is a primary lateral meristem? As I said, when you look at the dicot stem, the meristem which is already available, but for secondary growth, laterally in position, so the correct option is fascicular cambium. So, C is the correct option for this. A group of permanent cells which are similar in structure and function. Very easy answer. These are called simple tissues. Parenchyma position is always in the following region. It is a mechanical tissue. So, and mechanical layer is hypodermis. So, it is in the hypodermal region. Simple mechanical tissue with lignified secondary cell wall is sclerenchyma. Let me clarify one thing here. Parenchyma and meristem, they have primary cell wall composed of cellulose. Parenchyma is having the cell wall deposited with hemicellulose or pectin. So, it is pectinized cell wall. Xylem, trachea, tracheids along with sclerenchyma fibers or sclerites, they have thick walled deposition, that is lignin. So, lignified secondary walls are in sclerenchyma. The sieve cells are found in lower plants. In kingdom Plantae, algae are aquatic, they are thalloid. Bryophytes are thalloid but avascular. So, these two groups we do not find vascular tissues. Now, the remaining are pteridophytes, gymnosperms and angiosperms. While pteridophytes and gymnosperms, we call it as the primitive lower plants, and the well developed plants are angiosperms. So, sieve cells are in gymnosperms and pteridophytes. The correct option is C. Albuminous cells are found in the phloem of Funaria is a bryophyte, Selaginellite pteridophytes, pterids is pteridophytes, gymnosperms which are adapted for mostly the xeric conditions, and in gymnosperms along with the sieve tubes and companion cells, some parenchyma they store albumin. So, these albuminous cells are associated with the phloem in gymnospermae. The sclerenchymatous hypodermis is present in, definitely it is a monocot stem for giving mechanical support. In dorsiventral leaf, it is a dicot leaf where stomata are always towards the lower epidermis to prevent excess loss of water from the upper side. So, they are equally distributed. In dorsiventral leaf, the stomata are more on the lower epidermis. So, the correct option is B. The oldest layer of secondary phloem in dicot stem is located just outside the secondary xylem. So, this is the correct answer. After 2 years of secondary growth, the cortex in dicot root, as the stily part is increasing, there is no space available. So, it is completely soft away. Youngest layer of secondary xylem is always adjacent to pericycle. As a tree grows older, which increases rapidly in thickness, definitely is a heartwood or duramen. The function of hypodermis in Helianthus is giving hardness and rigidity. The radial vascular bundles are seen in all types of roots. Endodermis of roots with passage and Casparian strips, it prevents leakage of water from stele, and it also increases the pressure when there is a radial conduction of water. The chief conducting element in the phloem of angiosperm is sieve tubes, as sieve cells are in lower plants. The major part of ground tissue of the leaf is definitely a mesophyll. Major part of ground tissue of the leaf, yes, mesophyll. Which among the following is a geocarpic fruit? The fruit developing below soil is garlic. Vegetative propagation in Pistia, it is a hydrophyte and it is by offset. Periderm is the collective term for all the above. Phloem, phloem and phloderm. Seed coat is not thin, membranous in coconut. Roots play insignificant role in absorption of water in Pistia. Root hairs develop from the region of maturation. Proximal end of the filament of stamen is definitely attached to the thalamus. If you look at the two columns, cocambium coc, alburnum duramen, phloderm, these all are the layers which develops in dicot stem during secondary growth. Cocambium is called phloem. Cork is a phloem. Alburnum is a sapwood. duramen is a heartwood and phallodem is a secondary cortex. So, the correct option is B. Coming to the end of the session, assertion where you read in some flowers like lily the term perianth is used when calyx and corolla are not distinct, and reason calyx and corolla are the reproductive organs. Here assertion is correct but the reason is false. So, the correct option is C. Coming to end of the session, thank you very much. I know this is the vast topic. Okay, dear students, continuous reading, get the knowledge, ask explanation if you have any doubts, and clarify the doubts as and when it arises, and whenever any major doubts are continuously striking to your minds, don't worry, don't go in stress. Okay, have your own time and go through first morphology, then taxonomy including 3 families, then learn plant tissues, and then learn anatomy, and then slightly give little stress to secondary growth. Thank you very much.